Hi there, I'm Lisa Schimmold. Welcome to Microbiology at Crafton Hills College. What I'd like to do today is give you a little bit of an overview of what we're going to be covering this semester. I think you're going to find this material to be quite interesting. I know I still do, even after all these years of teaching microbiology. But let's go ahead and get started. Now, I hope you'll forgive me. You'll see me looking back and forth a little bit. I'm going to be looking at my notes and your lecture outline, and also the clock. I want to try to keep these videos to a maximum of about 15 minutes. Now, in order for this lecture to be user-friendly to you, you do need to go to Blackboard and print a copy of the outline for the chapter that I've provided for you there. Or you can just download it to your computer or your tablet and take notes into that. You will definitely need to take notes, detailed notes, and I'll make some comments as I move through this material. Okay, let's pick it up with bacteriology. And bacteriology is the study of the bacteria, which are also known as the prokaryotic organisms or prokaryotes. Prokaryotic means, the word literally means pre-nucleus, and it's a reference to the structure of the bacteria. These are unicellular organisms. They're very tiny, maybe range in size from 1 to 6 or 7 microns in length if they happen to be bacilli. 1 micron, just for a point of reference, equals 1/1,000,000th of a meter. So these guys are very, very small. As I mentioned a moment ago, they are unicellular. That means one cell is one complete and independent organism. Prokaryotic, as I said a minute ago, means pre-nucleus, and this is a reference to the structure of these very simple cells. While they have DNA, deoxyribonucleic acid, our main genetic molecule, their DNA is not separated from the rest of the cell by means of a nuclear envelope. So we have one circular molecule, large molecule DNA in each bacterium, and while it may be perhaps attached at a point or two to the cell membrane, there is no nuclear envelope separating it from the rest of the cell, and so that chromosome is just sort of free-floating in the cytoplasm. Now, in addition to not having a nuclear envelope, the prokaryotes of the bacteria lack other organelles as well. And I mean things like mitochondria and Golgi complex, centrioles, chloroplasts like we see in plants. So while most of these functions still occur in bacterial cells, production of ATP, for example, they are done mostly within the cytoplasm, in areas within the cytoplasm. Some reactions actually will begin with some enzymes that are located embedded in the plasma membrane, but we don't have those distinctly compartmentalized organelles that we see in what we call eukaryotic cells. So those are the prokaryotes of the bacteria. Now, we have 3 basic morphologies that we see with the bacteria. Round cells are referred to as cocci. Coccus, C-O-C-C-U-S, would be the singular form of the word. Cocci would be the plural form of the word. Rod-shaped or elongated cells are known as bacilli. And then we have a variety of bacterial cells that may be curved or bent or even twisted into a small spiral. And a number of forms there. We have curved bacterial cells that are called vibrios, twisted cells that are called spirilla, and then there is another variety of bacterial cells known as the spirochetes, and they are spiral-shaped cells with a structure known as an axial filament. And I'll discuss axial filaments in a later lecture. All right, so we've got the 3 basic morphologies: cocci, bacilli, spirilla, and other curved or bent cells. And some bacteria are pathogenic. Now, the word pathogenic means having the ability to cause disease. Fact of the matter is, the vast majority of bacteria are not pathogenic to humans. Most of them are benign. They live in the soil, for example, in water, associated with other plants or animals. a very small number of bacterial species are pathogenic or potentially pathogenic in humans. And we'll spend quite a bit of time talking about some of those as the semester progresses. Now, let's talk about classification of the bacteria or the prokaryotes. Now, when we look at classification of organisms, what we're doing is we're trying to apply some order to the great diversity of life that we see on planet Earth. There are literally tens of thousands of species, most of them being bacteria, that live on our planet with us. And so scientists called taxonomists, what they do is they have worked on arranging all of this diversity of life on Earth into some categories to try to just separate them up a little bit to deal with the large number of organisms, to also perhaps establish relationships between organisms, try to understand the evolutionary history of all kinds of organisms. And there are actually a number of systems of taxonomy or classification. There are many different opinions on how organisms should be divided into groups. We, this semester, are going to use what's called the three domain, four kingdom system of classification. All right, so domains, those are the largest of these groupings, these taxonomic groupings. And there are three domains in this system. There is one domain called Archaea. That is a domain of prokaryotic organisms. And domain Archaea lacks a substance in the cell wall known as peptidoglycan. More on that later. The second domain is called either Eubacteria or just plain old bacteria. When you put EU in front of a word, it means true, right? So domain Archaea, domain Eubacteria, and then the third domain is called Eukarya. Now that domain contains all known, anyways, eukaryotic organisms. Eukaryotic means having a true nucleus and other organelles like the ones I mentioned earlier, and we'll talk more about that in just a few minutes. Okay, let's take a look at your notes. All right, a little bit more about domain Archaea. Now, these organisms, as I mentioned earlier, do not have a substance known as peptidoglycan in their cell wall. Members of that other bacterial domain, Eubacteria, they do, for the most part anyways, have peptidoglycan, always exceptions. But back to domain Archaea, no peptidoglycan in their cell wall, and many of the members of this domain live in what we might consider to be extreme environments. And we've got three categories, and these are found in your outline on the first page. The first of these categories of the organisms that are members of domain Archaea would be the halophiles. Now, halo means salt, phile means lover, so these are the salt lovers. These are bacteria that live in highly saline environments, much saltier than the ocean. I mean places like the Great Salt Lake, Salton Sea here in Southern California. Perhaps they might be found growing in salt flats in very arid areas. Anyways, they not only can tolerate high concentrations of salt, they actually require them to maintain the integrity of their cell walls. All right, so the halophiles, that would be one of our categories of extremophiles in domain Archaea. Next are the methanogens, and these are organisms that decompose organic material. They produce methane gas and some other rather odoriferous metabolic waste products. You would find these methanogens living in swamps, for example, sewage treatment facilities. They would play a role in the recycling of nutrients and the breakdown of organic material in that kind of a situation. And then the third category of domain Archaea extremophiles would be the thermophiles, and that word of course means heat lovers. These are bacteria that can survive in extremely hot environments. For example, there are underwater volcanic vents in the ocean, and sometimes they outgas, and so we'll have very, very hot gas escaping into the ocean, usually high sulfur content. This would be the kind of extreme situation where a thermophile would thrive, and most other forms of life could not survive there. So anyways, those are members of domain Archaea. All right, let's move on to the next category. Remember, what I'm doing in this lecture is giving you kind of an overview of what we're going to be covering this semester. So next on my list would be nomenclature and taxonomy, and I've already mentioned it a little bit, but let's talk about nomenclature, and then we'll talk in a bit more detail about taxonomy or classification. Nomenclature is the naming of organisms. And by that I'm referring to applying scientific names to organisms. A scientific name has two parts. The first name is called the genus name. The genus name is always capitalized. The second name is the species name. That word is going to be written all in lowercase. And then we would individually underline both names. Or if you were word processing, you could, rather than underline, you would italicize the name. You really wouldn't do both. You would either underline or italicize the name. Okay, now let's take a look at an example of a scientific name and kind of break it apart a little bit and see what kind of information we might find in there. Sometimes scientific names really do contain some useful information. As we'll see in a moment when we take a look at the scientific name Staphylococcus aureus. Other times, scientific names are going to reflect the name of the individual that perhaps discovered them, like the name Escherichia coli, named after Theodore Escherich. That's who first identified that bacterium. In other cases, the scientific name might reflect where the organism was first identified, or there may be some other information, but sometimes a scientific name will provide some rather significant information about the organism. So let's take a look at the scientific name Staphylococcus aureus, and you've got this in your outline. As it says in your notes, this organism is both part of our normal flora, and it can also be, under the right circumstances, an opportunistic pathogen. Let's talk about those two things. Now, the term normal flora, that refers to microorganisms. Could be bacteria, could be fungi, could be protozoans in some cases. But anyways, these organisms colonize us. They colonize the surface of our body. Many of them are found living inside of our body, like in our upper respiratory tract, in our digestive system, urinary and reproductive systems, for example. And under normal conditions, this is perfectly healthy. As a matter of fact, you would not be healthy without your complement of normal flora. But in other cases, let's say if our immune system becomes compromised for any of a number of reasons, sick with something else, taking medication of any kind, steroids, for example, cigarette smoking, all of those and many, many other things can compromise our immune system from one degree to another. But anyway, when our immune system becomes compromised, our normal flora may become opportunistic. Now, an opportunistic pathogen, that's an organism that doesn't usually cause disease in humans unless the circumstances are favorable for that to happen. And Staphylococcus aureus, would be an example of normal flora that could be opportunistic. Now, Staph aureus, I said it was normal flora. Normal flora of the nasal caries, of the sinuses, found there in small numbers. There are many other microorganisms that are part of the normal flora of our upper respiratory system, and they compete with each other and keep the numbers of the other guy low, so they don't usually cause us any problems. But when the situation becomes unbalanced, Staphylococcus aureus can cause a number of problems. For example, if it contaminates food, from the sinuses or from the skin to food. Staph aureus produces a very potent enterotoxin. It's a very heat-stable toxin, and it causes, upon ingestion, the symptoms of a very common form of food poisoning called staphylococcal food poisoning. It causes maybe 25% of food poisoning cases a year. Have you ever heard the expression, the 24-hour flu? I bet you have. Well, that's really a misnomer, because there really is no such thing as a 24-hour flu. Typically, that is a case of food poisoning, staphylococcal food poisoning possibly, which usually resolves after about 24 hours of misery. All right, now Staph aureus can also cause a disease called toxic shock syndrome, and I will be talking about both of these diseases in more detail later this semester. But let's take a look at the name Staphylococcus aureus. First of all, if you look at the genus name Staphylococcus, we've got some information there. Cocci, well, we know that this organism is made of, or consists of round bacterial cells, cocci. Staphylo refers to arranged in clusters, sometimes referred to as grape-like clusters, and I think there's a diagram, if it's not in this chapter, then in the next chapter, showing you the arrangement, possible arrangements of bacterial cells. So that may not sound like a lot of information to you, round bacterial cells arranged in clusters, but actually it is significant. If a physician or a microbiologist is aware that a patient's infection is caused by a member of the genus Staphylococcus, they would have a pretty good idea of what antibiotic or antibiotics might be appropriate to treat that infection. So that's pretty important. Now, the species name aureus, it is derived from, I believe, Greek rather than Latin in this case, and it means golden. So sometimes we call Staph aureus the golden staph. And this is a reference to, if you grow Staph aureus on a type of microbiological media called tryptic soy agar, the colonies typically are just white, nothing unusual about them. But if we were to inoculate that same organism, Staph aureus, onto a type of agar called blood agar, after incubation we would see pale yellow colonies. And again, an experienced microbiologist could look at those and say, I have a feeling I'm dealing with Staph aureus here. So quite a bit of information in that name. All right, now taxonomy. You should have a handout entitled 3-Domain, 4-Kingdom System of Classification. We've already talked a little bit about 2 of the 3 domains, domain Archaea, domain Eubacteria, and then the 3rd domain is called Eukarya. Now, domain Eukarya can be broken down into smaller groups referred to as kingdoms. Let's talk just momentarily about those. First of all, kingdom fungi, pretty diverse group of organisms. We've got unicellular fungi, those are known as the yeast, and a number of multicellular fungi, for example, molds and mushrooms and plant pathogens called rusts and mildews, for example, and there are others. Very diverse group. The yeast, as I mentioned a second ago, are unicellular. All of the other forms of fungi that I mentioned after that are all multicellular. Now, what the fungi have in common is, in addition to being eukaryotic, they absorb their nutrients through their outer covering, which is sometimes called a cuticle. All right, next is kingdom Protista, also known as the protozoans. This would include things like euglena and amoeba, and then we're going to talk also about some pathogenic protozoans. These are eukaryotic organisms, and they are unicellular. Some are heterotrophic, meaning they are going to ingest preformed food. Others are autotrophic, meaning they have the ability to use a process called photosynthesis to synthesize their own nutrients. Okay, next is kingdom Plantae, multicellular, use photosynthesis, and these are what we call the plants. And then the 4th kingdom is kingdom Animalia. That's the kingdom that you're a member of. These are eukaryotic, multicellular organisms that ingest their nutrients, meaning they take their nutrients in through a specialized opening. Most of the time we refer to that as a mouth. Okay guys, I'm going to break about now. I'm trying to keep these videos at about 15 minutes or less. So take a look on YouTube and you'll find part 2 of this lecture in just a few minutes. Thanks for watching.